In this video, we will continue our discussion about the main topic, that is how to overcome challenges in the TME, that is tumor microenvironment. And these challenges overcome we are talking about with respect to the car development, or what are the modifications that one can do in the car design to improve their challenges in the TME and thereby increase the CAR T therapies efficiencies. So some of the points are like a repetition of what we have discussed in a previous video, that is T cell regulations. So in this case, the factors that helps in the T cell signalings or the activations needs to be upregulated, such as metabolic enzyme, uh, nutrient transporter, and this, for example, this glucose amino acid upregulations needs to be upregulated, and the nuclear factors also needs to be upregulated so that it can release more transcription factors. It can upregulate the transcription factors, and that helps in the T cells activations. At the same time, we need to suppress dominant negative or the switch receptors. So this dominant negative and switch receptors function is to suppress the T cells activations. For example, the TGF-β receptors that will suppress the T cells activations, and also these switch receptors. For example, this immune checkpoint inhibitors receptors. So whenever they get activated, they will suppress the immune response against the tumors. And this is one of the strategies used by the tumors to counter this immune response by the host. The other things that we need to do is uh, we need to suppress the cytokine receptors that will lead to the immunomodulations. And some of the ways to do is that by suppressing the receptors against the cytokines that cause immunomodulations, and at the same times we need to design our these antigens against the tumors to be as small as possible so that it does not create a tonic response on its own. The next target or next category is about a tumor visibility and targeting. This is one of the ingenious strategies that can be used by upregulating the non-encoding RNA in the extracellular vesicles, and this non-encoding RNAs will help in the visibility of the visibility against the tumor, and also helps in the targeting. And this bite is kind of a car with two antigens specific, or it will have the two informations about the two different antigens of the tumors. The next one is this ECM targeting enzymes, or the next one is this tumor infiltrations and trafficking. So in this case, the idea is that to improve the CAR T cells trafficking towards the tumors, and one of the receptors that is known for these functions, this trafficking, is the homing receptors. It helps the immune cells to reach to its site and get exit from the bloodstream. At the same time, when we reach to the tumor sites, because of the highly dense tumor microenvironment, which can be made of the fibers, in case of the pancreatic cancers, we need enzymes that can help in the penetrations of this TME and reach to the tumors. And this can be done by upregulating the ECM targeting enzymes. One of the example is this heparinase. Heparinase will helps in the degradations of this TME, and at the same time also helps this upregulation of these homing receptors will helps the tumors to reach to their will helps the CAR-Ts to reach to their final site, that is targeting the cancers. So these are four major categories that are described in the figures, and first two of them are kind of a repetitions of what we have discussed previously, but other two are some new advancements or the new dimensions of the design for the CAR-Ts that can help in the T cells, that can help in the this T cells anti-tumor efficacies, and ultimately helps or improves the CAR T-cell therapy against the solid tumors. So that's all for this video. If you need to understand more about the strategies in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.